Great, okay. If you wouldn't mind taking your seats, and we'll continue the same session, 3 additional speakers. Our first speaker is going to be Hannah Holt, Hazard Prioritization and Resource Scarce Settings, Case Study in Punjabi Dairy Industry in India.
Thank you. So I'm Hannah Holt. I'm a PhD student from the Royal Veterinary College. And my PhD was focusing on a specific zoonotic disease, so brucellosis in dairy cattle and humans in Punjab state of India. And this was kind of a spin-off from that. And it forms the first chapter of my thesis, which hasn't been submitted yet, so feel free to come give me feedback and criticism. It should be submitted soon, hopefully. So foodborne hazards, and by that I mean bacteria, viruses, parasites, toxins, chemicals, pose a vast global burden on public health. And policymakers require information in order to prioritize these hazards in terms of the risk to consumer. And this requires an understanding of the food systems in order to identify where hazards might enter and whether or not they might be retained in the final product. Raw milk may be contaminated with a wide range of hazards, so pathogens either from direct shedding in milk, fecal contamination, or contamination in the post-milking environment, so either at farm level or later in the stage. There's also concerns with chemical hazards, either via adulteration or unintentional, so for example via feed. India is the world's leading milk producer, if we're counting cow and buffalo milk, and Punjab state, where I was working, produces the most milk per capita. And despite the mass consumption of milk and dairy products in this area, there's been no formal assessment of the risks posed to public health from the consumption of this. So this was a risk ranking exercise in order to identify, based on what information was already available, priority public health hazards associated with consumption of cow and buffalo milk and dairy products in Punjab, to begin to identify possible critical control points and also key knowledge and data gaps precluding a formal assessment. So in terms of my PhD, this gave me a preliminary assessment of the relative importance of Brucella as a foodborne hazard, and also helped me to understand the structure of the dairy sector here. And this pilot exercise was also used to form future proposals to look at milk safety in India. So the hazard prioritization exercise contained several components. So first, we performed a situation analysis of the dairy industry, which data for this were obtained through a semi-structured literature review, expert elicitation, and consultation of the relevant legislation. And the data gathered fell under 3 themes, so a description of the producers, the supply chains, and the regulatory frameworks within which they exist, a summary of the information on possible hazards present at different stages in the dairy chain and factors influencing their presence, and then common processing and consumption practices in the study area. So as well as conducting an expert opinion workshop, we also visited a large dairy processing plant of a producer in the formal sector and interviewed key personnel regarding their procurement, processing, and milk testing practices, and looked at their SOPs regarding pasteurization and quality assurance. So this fed into a hazard identification. So during a working group meeting, we looked at decided which hazards could potentially be present in milk and dairy based on the evidence we had. I think the animation's a bit weird, but anyway. So there was not enough information to perform a formal formal exposure assessment within a traditional risk assessment framework. So we did what we called a rapid exposure assessment. So we summarized and assimilated all the available data on milk-borne hazards, and then we described the steps in the pathway from producer to consumer to discuss which were the risk amplifying and potential risk reducing steps. And here we're defining risk as a function of the likelihood exposure, the probability of infection or disease following exposure, and the severity of illness. So we performed a consequence assessment as well. So for pathogens, biological hazards, This was a combination of like morbidity and mortality expressed as disability adjusted life years per 1,000 cases, and also dose response information. So this was kind of taken a little bit adapted from Havla and some things that EFSA are using. And we combined this in a hazard prioritisation. So I will just take you through some of what we found. So the majority of milk here is still produced by smallholders and is sold in the informal channel. So that's either direct to consumers or to unlicensed milk vendors. There's no legislation regarding raw milk, but there is legislation regarding pasteurized milk and dairy products. There's room for improvement in milk hygiene at the level of the producer. So, for example, studies found that farmers were washing udders before milking, but just using water and not disinfection for the majority. And what we realized is there's a wide range of dairy products consumed in Punjab, so it's key to the diet here, and we had to consider the different processing methods of these. So for our hazard identification, we adapted a framework used by the European Food Standards Agency to see, based on the expert elicitation and the literature, what hazards we thought could be present, both biological and chemical. So these are some of the results. So that's a list of our pathogens and biological hazards that we took forward to the next step. And then in terms of chemicals, we also looked at like pesticides, aflatoxins, adulterates, antimicrobials. It's important to say that these were the hazards for which we had some evidence of presence, but for any of the potential hazards that you can find in milk, we didn't have any evidence of absence. So although we didn't take these forward to the next stage, we can't exclude anything based on what data is already available. So I won't go through these tables in detail, but they're just an example of some of the results that we found from the literature review. So the most common pathogens we found were E. coli and Staph aureus, and as well as raw milk and products produced in the informal sector, these pathogens have also been detected in pasteurized milk. In terms of chemical hazards, pesticides had been investigated the most, and these have been found above maximum recommended levels in both the informal sector and in pasteurized milk produced by the formal sector. This is just a summary of some of the risk amplifying and risk reducing steps that we found on the pathway from producer to consumer. So evidence suggests that adulteration with water, urea, detergent, sodium hydroxide, even formalin is common practice, and this can occur at the stage of the producer or intermediary, so milk vendors. There's no on-farm refrigeration or refrigeration in the formal sector, which could reduce in pathogen proliferation, but milk is generally sold quite quickly through this channel. So there's no screening for milk. In terms of the formal sector, because we've found E. coli and Staph aureus, and at the point of retail, this milk has been found to have standard plate counts above the legislated dose. So this suggests that either there's some problems with pasteurization or that post-pasteurization contamination can occur. And in terms of so consumers are boiling milk in general, and the majority of milk is heat treated, which will reduce consumer exposure to biological hazards. Although there's some products like sweets made from milk that consumers would not heat treat that have been found to contain pathogens. And also these chemical hazards that we're discussing are not altered by pasteurization or consumer processing. So I won't go through this in detail, but this is kind of a summary of the types of things that we were producing. So in terms of the biological hazards that we took forward, we felt that any of them could be present at the point of retail, although for some of these pathogens like Brucella and Cryptosporidium, they've only been looked at in terms of being present in dairy herds in the animals, and we haven't looked at whether they can be present in milk and dairy products. As well as presenting this, we also looked at the strength of evidence. So we evaluated biases from the studies we included, and we found that the majority of pathogens had weak or very weak evidence. So this needs to be looked at further, and that a lot of processing that's done at consumer level may reduce the impact. So just to summarize, for biological hazards, when we combined consequence with exposure, we found that the hazards most likely to be present are those for which post-processing contamination is possible. So we prioritized E. coli and Listeria, with Campylobacter and Salmonella needing further work. So I'm out of time. But yeah, we also found that evidence of presence of chemical adulterates, aflatoxins, and pesticides, and these, as I mentioned, won't be inactivated by processing. So we felt that due to processing and consumption practices, there's probably a higher risk of exposure to chemical hazards, but at this stage we can't consider that any hazards have a negligible risk, and this is definitely something that we should look into further. And this assessment was used to inform future work, so we're now starting a new project funded by the Bill and Melinda Gates Foundation and DFID to take a systems approach to milk safety instead of focusing on a particular pathogen or aspect of the problem. So come talk to me if you want to know about that.